everyone, welcome back to the lecture series session of overview and integration of cellular metabolism. So, we were in the discussion of fatty acid catabolism, mainly the oxidations of fatty acids. So, the topics which we already have covered are the different types of fatty, in the different types of fatty oxidation we have vividly discussed beta oxidation of fatty acids, the process of beta oxidation starting from activation to transport of fatty acid inside mitochondria, then the different reactionary steps of all the different types of fatty acids like ORT chain, even chain fatty acids, then saturated, unsaturated fatty acids. Then we have discussed the energetics of beta oxidation, regulation and also the related disorders. Now, in this class we are going to discuss other types of fatty acid oxidations like alpha oxidation, omega oxidation and peroxisomal oxidation of fatty acids. Now, if you remember from the previous classes that alpha oxidations and omega oxidations are based on the numbers of carbons in fatty acid. So, if you remember that omega carbon is the distant most carbon from the carboxylic group in fatty acid, whereas the carbons which are attached to fatty carboxylic group of fatty acids, the first one is known as alpha, the second one is known as beta. Based on that, these the carbons are named, as well as when those specific carbons like alpha carbons, beta carbons or omega carbons are oxidized, that type of fatty acid oxidation is named accordingly. Now, peroxisomal oxidation is named based on where the oxidation of fatty acid is occurring. So, as we have discussed that beta oxidation of fatty acid, the organelle for beta oxidation is mitochondria, whereas peroxisomal oxidation occurs in peroxisome. Now, peroxisome is one membrane bound organelle present in both plants and animals, and this organelle is very much known for the presence of the enzyme catalase. It is a this peroxisome is a catalase rich organelle, and also it is dealing with see, there are different type of oxidations like amino acid oxidation, fatty acid oxidation, those are occurring in peroxisome, from which free radicals are generated, and those free radicals are rather neutralized with the help of the enzyme catalase. So, peroxisome is one such very known organelle for handling different free radicals. Now, peroxisomal oxidation, the target fatty acids are mostly either very long chain fatty acid or branch chain fatty acids, and those fatty acids are mostly obtained from dairy products, fats of ruminant animals, meat, fish. These are the origin of very long chain or branch chain fatty acids. Now, peroxisomal oxidation is more or less similar with the beta oxidation which occurs in mitochondria, and it generally peroxisomal oxidation also consists that same 4 steps, that is dehydrogenation, then hydration or addition of water, then finally oxidation and then thiolytic cleavage. Now, then where is the difference? The difference lies in 2 specific conditions. One is, and that those conditions are related to the first step only. Now, the first step, the enzyme is a flavoprotein dependent oxidase here in peroxisomal oxidation, and that flavoprotein dependent acyl coenzyme oxidase is transferring the electron directly to hydrogen peroxide. Now, in case of beta oxidation, this is our beta oxidation shown here. So, if you remember, the electron through the flavoproteins in beta oxidation is transferred to electron transferring flavoprotein which carries the electron to mitochondria electron transport chain. But in case of peroxisomal oxidation, these electron does not enter ETC, because definitely this is peroxisome. So, what happens to those electrons? They are directly transferred to H2O2, and these H2O2, actually they are directly transferred to molecular oxygen forming H2O2, and this H2O2 is broken down with the help of the enzyme catalase. So, basically here no ATP is formed. So, no ATP from this stage, rather the energy released is basically dissipated as heat. So, in the first step there is even if there is electron transfer through flavoprotein, those electrons does not enter ETC, rather it forms H2O2, which is degraded with the help of the enzyme catalase, and the energy is released as heat. Next, the specificity of this acyl coenzyme oxidase also differs. They prefer these enzymes prefer very long chain fatty acids like hexacosanoic acid, that is a 26 carbon fatty acid, and also branch chain fatty acids like phytanic acid or pristanic acid. So, the specificity of this enzyme, the enzyme designated for the first step, that is fatty acyl coenzyme oxidase, its specificity as well as the way of transferring electron differ with the beta oxidation of fatty acid. So, this is peroxisomal oxidation of fatty acid. Now, remember peroxisomal fatty acid, the oxidation occurs in peroxisome where there is no TCA cycle. Now, when there is excess branch chain or very long chain fatty acid ingestion, there is high concentration of this type of fat in our diet, what it does, it has some effect over peroxisomal beta oxidation located in liver. So, the enzymes related to peroxisomal beta oxidations are basically increased. Now, what happens, this peroxisome does not contain TCA cycle. So, what will be the fate of those fatty acids broken down? So, basically in liver they form shorter chain fatty acid products like hexanoyl coenzyme, and those shorter chain fatty acid comes out of peroxisome and enters in mitochondria to follow the complete degradation or complete oxidation through beta oxidation to form acetyl coenzyme A and carbon dioxide. So, this is peroxisomal oxidation. Peroxisomal oxidation takes help of mitochondrial beta oxidation as well for the complete oxidation of the long chain or branch chain fatty acids. Now, we are going to discuss few disorders related to peroxisomal oxidation. Now, one such disorder is Zellweger syndrome. Now, Zellweger syndrome is related to absence or reduced peroxisome in cell. So, either there is peroxisomal content is reduced or there is complete absence of peroxisome. Now, what happens, there are few genes, PEX genes basically. Those PEX genes are synthesizing a specific protein which causes organization of peroxisome. Now, because of the deficiency of that specific protein which is also known as peroxine, this peroxine protein deficiency causes disorganized or formation of peroxisome. Sometimes peroxisome is completely absent. Now, what will be the effect? All the metabolic pathways which happens in peroxisome, they will be absent. So, including peroxisomal fatty acid oxidation, it will also be hampered. Then there is another disease, adrenoleukodystrophy. Sometimes we call it X-linked adrenoleukodystrophy because the defect or the mutation is related to the X chromosome. Now, the defect lies in a peroxisomal membrane transporter. Those membrane transporter proteins located in peroxisome is mostly carrying the branched or long chain fatty acids. So, the tissues which are mostly affected are central nervous system, where those long chain or branch chain fatty acids are synthesizing the myelin sheath. So, basically myelin sheath formation is affected, then adrenal cortex affected, Leydig cells of testis also affected. So, basically what is the fate that the long chain fatty acids accumulated, those long chain fatty acid causes damage to the myelin sheaths. Now, XALD or X-linked adrenoleukodystrophy, they affects young boys, and the manifestation mostly occurs before 10 years of age. There are different types of neuronal manifestation, loss of vision, behavioral disturbances, and finally they can lead to death as well. So, in both of these diseases, basically there is peroxisomal fatty acid oxidation is affected, leading to accumulation of very long chain fatty acids in the circulation, and their detection in blood can be a screening for diagnosing these diseases. Next, we will move on to alpha oxidation. So, alpha oxidation, here we are talking about oxidation of the alpha carbon of fatty acid. So, mostly this alpha oxidation is related to branch chain fatty acid, where the single carbon is removed from carboxylane, the just adjacent carbon to the carboxyl carbon is removed via oxidation. Now, again the steps in this alpha oxidation, important one is hydroxylation of the alpha carbon, then that is finally oxidized to alpha keto acid. Again the alpha oxidation occurs mostly in peroxisome, and the important organ here is brain. Then keto acid follows decarboxylation, releasing one molecule of carbon dioxide, which forms a new fatty acid which is one carbon less. Now, why this alpha oxidation is there? Why branch chain fatty acids mostly undergo alpha oxidation? Because there are few dietary branch chain fatty acids which have methyl group in the beta carbon. So, basically those beta carbon cannot undergo beta oxidation because that cannot be oxidized. So, what we need is to remove this hydroxyl group from alpha carbon. Now, one such important dietary methylated fatty acid is phytanic acid. So, here we will discuss the alpha oxidation with the example of phytanic acid. So, here you can see that the alpha in the beta carbon there is hydroxyl group present. So, with the help of the enzyme phytanoyl synthetase, we get the activated fatty acid, that is phytanoyl coenzyme A. Then comes the important enzyme, that is phytanoyl coenzyme A hydroxylase. Now, with this hydroxylase enzyme, what happens? Here you can see there is decarboxylation. So, one new fatty acyl coenzyme A is formed, which is one carbon less. Then we are getting hydroxyphytanoyl coenzyme A. So, this alpha carbon here you can see is hydroxylated. So, with the help of the hydroxylase enzyme, there is decarboxylation as well as hydroxylation of the alpha carbon. Next, with the help of the enzyme lyase, formyl coenzyme A is released, forming pristanic acid or pristanol. This is the aldehyde form, pristanol. Now, this pristanic acid further can undergo beta oxidation via forming pristanic acid. This pristanic acid then can enter the proper beta oxidation. So, once again in alpha oxidation what happens, the problem is there is a methyl group in beta carbon position for which beta oxidation cannot occur. So, what is done is the first thing is alpha carbon is hydroxylated as well as there is decarboxylation. Then this part is removed as formyl coenzyme A, forming pristanol. Pristanol is forming pristanic acid. Now, pristanic acid is basically derived from the phytanic acid only, and then pristanic acid undergoes the normal beta oxidation after entering in mitochondria. So, the end product which is generated from the oxidation of pristanic acid is propionyl coenzyme A form, and this part which is trimethyl decanoyl coenzyme A. So, this is alpha oxidation. Now, there are few one defect related to alpha oxidation. One important one is Riftsums disease. Now, Riftsums disease is related to that specific enzyme which is exclusive in alpha oxidation, that is alpha hydroxylase enzyme, that is the phytanic acid oxidase enzyme. Now, what happens, definitely the phytanic acid oxidation is hampered, leading to phytanic acid accumulation, and this phytanic acid affects the central nervous system as well as peripheral nervous system. So, what we can see, there is polyneuropathy, retinitis pigmentosa, nerve deafness as well as cerebellar ataxia. Now, phytanic acid mostly is not endogenously derived acid. So, basically that is the phytanic acid is obtained from diet. So, as therapeutic regimen, what should be done is to restrict phytanic acid containing food, and phytanic acid mostly present in the fish, milk, fats from ruminant animals, tuna, cods, these type of fish, they contain phytanic acid. So, these diets should be avoided. Along with that, accumulated phytanic acid can be cleared from circulation via plasmapheresis. So, this is Riftsums disease, but the other variant of Riftsums disease is infantile Riftsums disease. Now, infantile Riftsums disease is a condition where children are affected at the very early age, and their manifestations are mostly like Zellweger syndrome, where basically the peroxisome synthesis or peroxisome formation is hampered, leading to alpha oxidation defect. Then we are coming to omega oxidation. Now, once again, omega carbon is the distant most carbon from the carboxyl group in the fatty acid. So, it is just opposite direction of beta oxidation or alpha oxidation, where the omega oxidation is occurring in the carbon chain. Now, omega oxidation is located in endoplasmic reticulum, and this occurs mostly in liver as well as kidney. Now, the preferred substrates are the fatty acids with at least 10 to 12 carbon chains. Then omega oxidation, remember omega oxidation is the minor pathway, and in normal persons there is omega oxidation along with the major fatty acid oxidation pathway, that is beta oxidation. But omega oxidation, the manifestation or the omega oxidation process is exaggerated when the beta oxidation is defective. And what is the fate? There is dicarboxylic aciduria. So, how this dicarboxylic aciduria occurs when there is beta oxidation defective, we will discuss via the enzymatic reactions of omega oxidation. So, here you can see the fatty acids. This is our omega carbon, which is distant most from the carboxylic group in the fatty acid molecule. Now, the most important enzyme in omega oxidation is the hydroxylase enzyme, that is a mixed function oxidase. Now, with the help of this mixed function oxidase and NADPH, there is formation of an alcohol. So, this methyl carbon is converted to methyl alcohol with the help of mixed function oxidase. Then there are few dehydrogenases, that is alcohol dehydrogenase, which convert this alcohol group to aldehyde. Then aldehyde dehydrogenase, which convert this aldehyde group to acid group, rather carboxylic acid group. So, here after this mixed function oxidase and subsequent dehydrogenase treatment, what we are getting? A carboxyl group formation in the omega carbon, as well as the previously or the inherent carboxyl group which was there in the fatty acid. So, basically we are getting a dicarboxylic acid. Now, this dicarboxylic acid can be activated from the both side. So, these carboxyl groups on the both side can be activated, forming coenzyme A or acyl derivative of fatty acid, and this enters mitochondria for further beta oxidation. So, remember, omega oxidation occurs in the omega carbon only for single time, forming a dicarboxylic acid. Then that dicarboxylic acid actually enters mitochondria for further beta oxidation. Now, just assume the situation where beta oxidation is defective. So, what will be the fate of these fatty acids? They will form dicarboxylic acid, but there will be no beta oxidation. So, these dicarboxylic acids will be excreted in urine. That is why in defective beta oxidation there is dicarboxylic aciduria. So, what is the fate of the beta oxidation of these dicarboxylic acids? There is formation of succinate, the end products, and adipic acid. So, these are the two end products of beta oxidation of those dicarboxylic fatty acids which were originated in the process of omega oxidation. Remember, that is one very important question. So, again, if there is beta oxidation defects in the enzymes dehydrogenases or thiolases or lyases in ketone body formation, in those cases there is dicarboxylic aciduria as well as non-ketotic hypoglycemia and also fatty liver. So, these are the key points of today's class that we have discussed. Alpha oxidation. Alpha oxidation is specific for the branch chain amino acid where there is a methyl group in beta carbon. In our dietary, the fatty acid which we get from diet is phytanic acid, which is a branch chain methylated fatty acid. for which there is alpha oxidation and also the alpha oxidation defect, Riftsums disease or infantile Riftsums disease we have discussed. Then we have discussed peroxisomal oxidation, which is specific for either very long chain fatty acids or branch chain fatty acids. The peroxisomal oxidation is similar to the mitochondrial oxidation, but the difference is in the first oxidation step, where the transfer of electron occurs directly to the molecular oxygen forming H2O2, which is degraded, dissipating the energy as heat. Then we discussed omega oxidation, which occurs in endoplasmic reticulum, and there is formation of dicarboxylic fatty acid, and that intermediate, remember the once again that the product of omega oxidation is a dicarboxylic fatty acid, which enters mitochondria for further breakdown through beta oxidation. So, these are my references, and see you in the next class. Thank you.